Shall I start? Okay, I will start with the introduction about the Living Agro project. Okay, so thank you all for being here, especially to Professor Micheli and Dr. Ofensu for their availability to speak today in this technology dissemination workshop. It is among the many events that the project Living Agro is organizing to spread knowledge about available technologies in the agroforestry field. I will say just a few words of the Living Agro project so that you can see where this workshop stands. It's a project that lasts 4 years. Its title is Cross-Border Living Laboratories for Agroforestry. It's funded by the NECBC Med program under the Technology Transfer Program Priority. Where is the cross-border dimension? There is a cooperation between Greece, Italy on the one hand, and Lebanon and Jordan for the Middle East area, with several partners involved, and you can see them here. The second term of the project, Cross-Border Living Laboratories, is this new word, living laboratory, which is quite, broadly used now in many European projects because it is important to involve end users and real-life settings into research projects. So the key features of a living laboratories is just a network of people, a network of stakeholders in an open innovation ecosystem that try to integrate the research and innovation with a community, within a community of public-private stakeholders. The key features are the real-life setting, so we have pilot trials, user-centered, so we ask end users what they need in terms of innovation, and this co-creation approach. So Living Agro, Living Labs are just active collaborations between farmers, industry, local authorities, researchers to try to deliver these innovations in the field. The field in case is the agroforestry, which is the land use system that combines trees and crops or trees and animals on the same land unit. And depending on this combination, we have very varied systems, multifunctional systems. They can be sylvo-pastoral systems. They can be of several types, as you see in this screen, in this image by Burgess and Rosati. Living Agro, Living Labs focus on two types of agroforestry systems. One is multifunctional olive systems, and one is grazed woodlands systems. They're both typical of Mediterranean area, and they both deliver lots of ecosystem services besides the productive functions. Regarding the co-creation approach, we engage with many people from all the countries we've been visiting: Lebanon, Jordan, Greece, and Italy. We had meetings, about 20 meetings with privates, and they told us what was needed and the innovations ideas they had. We run several pilots in all countries. These are a few images of firms we've involved in the trials, both in the field or in nurseries. And we listed the type of innovations that we would like to disseminate in the Living Agro project by developing catalogs, 10 catalogs of innovations that have been used during brokerage events that we carried out in Greece, Lebanon, and Jordan. No brokerage events in Italy were foreseen in the project. Here we had one-to-one meeting with enterprises, and we found what were really the interests regarding the innovations that we proposed. To join this network of Living Lab, you just need to log on to the ICT platform and use the available information and services that you will find there. It's a platform where you will find besides the catalogs, information about the field trials, some e-learning modules on agroforestry, and other fact sheets, news, also a forum where you can ask or receive ask questions and receive answers by key experts in the field, or contact directly these experts for any query you should have on a specific topic. There are also online training courses and tools such as pre-competitive analysis tool to test the feasibility of your innovation. So these are the channels where you can reach the Living Agro project, the official page, but the best one is, well, the social medias and the ICT platform. So this is the Living Agro project in a nutshell. If you want to know anything more about it, just contact us at this address. So now we can start with the key presentations of today. So I will introduce Dr. Safa Ofensu from the University of Sassari and the Research Center for Desertification in Italy. Please, Safa, the floor is yours.
Sarah,
yes?
If I may, it would be maybe better to introduce the session by the speech by Chiara Cesaracciu on Salamed project.
Oh, okay.
A brief introduction on the Salamed project, so that afterwards she can leave if she needs to do other things. And
my fault.
We can proceed.
Thank you. Thank you.
Chiara,
my fault, yes, I missed the list. So the floor is yours, Chiara, for the presentation on the Salamed project.
No problem. Wait, let me share my screen. Yes, so I will just give a brief introduction about the Salamed project and some examples, some insights from the way we are implementing the Living Lab approach in the different case studies and particularly in Sardinia. So just as a premise of the assumptions that that Salamed was based on, land degradation and desertification in Mediterranean socio-ecological systems is the result of the interaction between ecological but also socioeconomic processes. And these dynamics are threatening not only ecosystem conservation, but also the livelihoods of rural communities. So land degradation and desertification can be framed as a so-called wicked problem, and therefore it requires a systemic, multi-stakeholder and transdisciplinary approach. The challenge, one of the elements of complexity, is also represented by the fact that the Mediterranean is a mosaic of different contexts, and so we need tailored solutions that are context specific. So in a way, we need to move away from one-size-fits-all solutions and towards identifying a set of options to manage complexity in an evolving way. So these were some of the assumptions that inspired the project, which is funded by PRIMA and aims to identify, test, and validate tailored nature-based solutions, both to enhance the resilience of endangered Mediterranean drylands or to restore degraded ecosystems. Another objective of the project is to engage stakeholders, as you were saying, in living labs, not only to identify practical solutions for sustainable land and water management, but also to promote business opportunities that are associated to the sustainable management of land and water resources. So the project includes 15 partners that are located in both the northern and southern shores of the Mediterranean. There are 6 living labs located in hotspots for land degradation. The project started in April 2022, and it will have a duration of 3 years. So as I was saying, the project is based on a living lab approach, which, as you mentioned, is a very broadly used concept that was developed in urban settings, but is increasingly being recognized as a valuable approach to address sustainability challenges also in rural contexts. Within Salamed, we are trying to design living labs as laboratories, so they are informed by scientific knowledge, by the outcomes of field and lab experiments, but also as living systems. So they are rooted in a dynamic and long-lasting stakeholder engagement process. So in the living labs, stakeholders and researchers are interacting in a co-researching process that aims to test and validate practical solutions, while also removing barriers for innovation, promoting the scaling out of these practical solutions, and identifying business opportunities that are closely linked with sustainable and water management solutions. The living labs in Salamed focus on both dryland restoration and dryland resilience, and these are some examples of what we're doing in the southern Mediterranean. So, for example, in Tunisia, the living lab is focusing on the scaling out of managed aquifer recharge for sustainable agriculture. In Egypt, we're focusing on water harvesting, and in Morocco on argan forest restoration by testing subsurface water retention technologies. On the other hand, we have the living labs which are more focused on dryland resilience. And for example, in Sardinia, we are focusing on increasing the resilience of sylvo-pastoral systems. Of course, I will not go over in detail about all the case studies, but just some insights from the Sardinian Living Lab, which is located in Berchidda Monti, and it includes large-scale grazing systems and a mosaic of croplands, permanent grasslands, woodlands, and wooded grasslands. There are many challenges that are being tackled in this living lab, including the lack of oak regeneration and the increased spread of pathogens. But the ecosystem is threatened also by socioeconomic dynamics, as the limited economic profitability is driving and contributing to abandonment, and which in turn is constraining generational turnover and innovation within these systems. So the research group focused on developing a stakeholder map and also co-designing field and lab experiments with local stakeholders. In particular, focusing on evaluating the effectiveness of microbial-based technologies to enhance plant resilience to biotic and abiotic stressors, but also on monitoring the impact of sparse trees or woodlands on the hydrological balance. So the interviews that were carried out with local stakeholders in the previous months highlighted that there's an imbalance between the capacity of stakeholders to respond in an adaptive way to different pressures. And in particular, on the one hand, vineyard growers and dairy factory owners were identified as the most influential stakeholders, while shepherds, beef cattle farmers, and sheep breeders as the least influential and adaptive. An interesting thing that emerged is also that the range of ecosystem services perceived as associated with sylvo-pastoral systems is very diverse, but these ecosystem services are mostly taken for granted, and in particular, most influential stakeholders are unaware of the benefits that they receive in terms of ecosystem services from sylvo-pastoral farms. So what we are working on in the Living Lab is actually to trying to influence these biased perceptions, and by uncovering these connections between economic productivity and ecosystem resilience as an incentive to promote open innovation. So to conclude, understanding the sylvo-pastoral systems as learning systems can contribute to increasing their resilience, prevent their abandonment, and in turn preserve biodiversity and ecosystem services. We believe that if well designed as learning spaces, Living Labs can increase the capacity of stakeholders to perceive these interdependencies among them, and also to understand the coevolutionary dynamics between socioeconomic and biophysical processes. And we believe that these this continuous interaction in the Living Lab can also lead to the identification of business opportunities that are associated with soil and biodiversity conservation, as well as the valorization of ecosystem services. So this, in a way, is an invite to all the people listening to reach out, as we want to create synergies by building a common Living Lab for promoting systemic innovation of Mediterranean sylvo-pastoral systems. Thank you.
Thank you, Dr. Chiara Cesaraccio. I will leave the floor to Salva Ofensu from University of Sassari.
Thank you, Sarah, for the introduction. Can you see my screen?
Yes, we can.
Okay. So first of all, I'm Safa Ofensu from a junior researcher from the University of Sassari. I get my engineering, agricultural engineering degree from the University of Carthage in Tunisia, from Institut National Agronomique de Tunisie. I get my PhD at the plant pathology section at the University of Sassari. So today we will talk about trichoderma and ways to cope with biotic and abiotic stress in semi-arid sylvo-pastoral system, which is a part of the activities of the work package 2 of SalamMed project, dealing with technological management and monitoring options for land resilience and restoration. The main objective of the task we are leading is to select and test microorganisms beneficial for plant and soil biodiversity, in order to with the aim to increase or preserve the soil fertility, promoting a sustainable agriculture. In fact, the use of these beneficial microbes that are inhabit the rhizosphere are known to help the plant I'm sorry, to help the plant to resist to many type of stress that like the water or drought stress and the salt and stress, and could be a promising alternative to the traditional or chemical-based approach, especially in a climate change scenario. So Trichoderma is one of the most prevalent fungi present in the rhizosphere that can be found in all type of ecological diversity. They are ubiquitous, fast-growing, cosmopolitan, and widely distributed in many types of soil, including agricultural soil, forest, pasture land, and also desert soil, from cool to temperate, to tropical climate. These fungi are opportunistic, virulent, plant symbiont, as well as being parasites of other fungi. So, Trichoderma was first described 200 years ago in 1700 in Germany, and till now 75 species have been identified, the majority of which are considered as imported biocontrol agent. These include Trichoderma arsianum, Trichoderma coningi, Trichoderma virens, and Trichoderma amatum. Actually, a recent study has shown or has identified 4 new species of Trichoderma under the clade of Trichoderma arsianum, as you can see here, based on 3 sequences, partial gene sequences dataset that are ITS, RPP2, and the translation elongation factor. So characteristically, the fungus is identified through rapid growth, a brine green to yellow colored conidia, and with a branched conidiophore. So since the first recognized application of Trichoderma species as a biocontrol agent, especially against Rhizoctonia solani, followed by the discovery of gliotaxin as the third antimicrobial produced by Trichoderma, they have been widely applied for the management of many plant pathogens and associated diseases. Trichoderma operated through a mixed mode of action by using more than one mechanism of action, through a direct antagonistic interaction, such as competition, antibiosis, mycoparasitism, or through a complex and direct mechanism by stimulating induces systemic resistant desolubilization and the sequestration of the nutrient uptake, and the enhancement of plant growth. Trichoderma is also extensively used as a model organism to understand biological interaction among antagonistic fungi, host defense responses, and a plethora of heterologous proteins affecting plant metabolism and physiology. Here is the plant pathogen interaction map. And we can say that Trichoderma pathogen interaction take several forms through the action of its diverse secretum containing VOX, volatile organic compound, cell wall degrading enzyme, reactive oxygen species, the called DROS, and antimicrobial secondary metabolite. And by forming these interactions, Trichoderma increase plant fitness and tolerance against biotic stress, either by priming plant defense, increasing plant growth, or releasing pathogen pressure, leading to the enhanced growth responses. Similarly to Trichoderma fungi, other rhizosphere microorganisms like plant growth promoting bacteria and mycorrhizal fungi can establish beneficial interactions with plants, promoting plant growth and development, increasing the plant defense system and enhancing tolerance to different types of stresses. So the microbiome consortium of efficient strains for biological control helps in improving microbial efficacy under diverse soil and environmental conditions, maintaining the sustainability of the systems. Trichoderma and other root colonizing fungi infect roots such as mycorrhiza, which are fungal roots, are usually not plant pathogens. Their role consists in simply enhancing nutrient and water uptake. As you can see here, a common mycorrhizal network can be established through the connection of several plants through the mycelium of one or more fungi. Two types of mycorrhiza exist in the nature. Ectomycorrhiza, which that don't penetrate the plant root cortical cell, but it grows between the cell, and the endomycorrhiza that are not visible and penetrate the plant root cortical cell. Okay. But let me say before that the interaction of trichoderma with other microorganisms is not always neutral or synergistic. For example, there could be a competition against mycorrhiza because trichoderma produce many enzymes such as chitinase, glucanase, and so on. Now let's start to talk about some research about the use of trichoderma as antagonistic or biocontrol agent, or as a plant growth promotion microorganism. So in this work, the promotional effect of a species called Trichoderma gisuens coating on watermelon and mice crop has been tested. Results show that the germination, as you can see here, the germination rate of coated seeds were significantly increased for maize and for watermelon as well. Seed coating was also found to benefit plant growth as plant height, the root and stem, the root length and stem diameter, and the biomass production were significantly higher than in the control group. This conceptual diagram illustrates the coating seed with Trichoderma gisuens can promote plant growth and help to prevent plant disease. It shows that the direct and indirect pathway through the microbial community that affect plant physiological entities, and we can see here the red line that indicate positive links. Another study about the use of Trichoderma arsianum for a biological control of turf grass. This study shows the direct and the use of Trichoderma arsianum. In this work, they tested the Trichoderma arsianum for its ability to control brown patch caused by Rhizoctonia solani, dollar spot caused by Sclerotinia species, and pythium root and rot and blight. So here the Trichoderma arseanum reduced the initial disease severity by almost 70% and delayed disease development by up to 30 days. The persistence of strain in soil for sample from treated creeping bentgrass greens was also measured. After application of this strain, soil population of Trichoderma increased 100-fold relative to population in untreated plots. And the population levels remained much more greater than in treated plots than in untreated, even after overwintering. Here, since not all Trichoderma strains protect plants to the same extent from water stress or drought stress, careful selection, including plant trials and the measurement of biochemical and molecular parameters in both the fungus and the plant is needed. In this survey, Trichoderma asperellum was the most promising strain out of 8 Trichoderma strain tested. Molecular and biochemical data from Trichoderma asperellum primate seedling indicate that they suffered less from the severe water stress applied compared to those untreated, which was supported by the down-regulation of the water stress response genes, the low antioxidant machinery, and the drop in the H2O2 content. Trichoderma atroviride was tested or used in another research and revealed promising results in increasing plant biomass and has overcame salinity stresses. This survey highlighted how Trichoderma asperellum serves as a potential source for establishment of vegetation. Work here in another work, we used Trichoderma asperellum to test the ability of this strain as a potential for establishment of vegetation or plant on red mood contaminated soils. What is the red mood? This is a sodic solid byproduct released by alumina and industry. So here, Trichoderma asperellum inoculation significantly lowered the pH and the electrical conductivity of the red mood, and it was able to improve growth, chlorophyll content, and reduce oxidative stress of Sudan grass. Last but not least, here I would like to give you some idea of about our experiments, about the effect of several species of Trichoderma on tomato plant infected by Pyronocheta lycopersici. As you can see here, there is a high similarity between the healthy root and the root treated with Trichoderma. Concluding, we can see that due its eco-friendly nature, Trichoderma is definitely good substitute to commercial synthetic fungicide in order to promote a sustainable agriculture. And thank you for your attention.
Thank you, Dr. Ofensu. Very, very interesting presentation. I wonder if there are questions from the audience by Mr. Kanari or Mr. Safi Ahmed. Please, if you have any question, you can just raise your hand or use the chat function, and we will see it there. If I may, just to start, I have a question. What did you use a coating from a commercial formulation, which is already available in the market, or are you using a coating that you do that will then become a commercial formulation within the Salamed project, as Ms. Cesaraccio explained?
Actually, now we are testing spore suspension conidia to find potential strains able to promote plant growth. But maybe Professor Migeli will present in his work about a formulation that is a combination between two strains that was identified by the same group, the group of Professor Migeli, and was the object of a commercial formulation. But I will leave the floor to Professor Migeli to explain more this topic. Thank you.
Okay, sorry. So I went too far ahead with the questions.
Not a problem.
Actually, the two presentations are linked together, so we just decided to split in two parts, and I will present the second part.
Okay, we can dive directly into your presentation.
Yeah, it should be, Safed needs to interrupt the sharing, so I can share myself. Okay.
Again, should there be questions from the registered users, you can use the chat function and we can answer from there, or you can raise your hands and just pose the question directly to Professor Migeli or the other speakers.
Of course.
Feel free.
So I don't know whether I cannot see any hand because I'm presenting in full screen, so
there are no hands raised so far, I think. Yes.
Okay. So besides the fact that okay, now it's visible. The second part of this presentation is more focused on the field applications and on the formulation. So the phases that are following the selection of active string from the bench, from the laboratory bench to the market and to the field application. Trichoderma is prone to a wide array of formulations, both solid formulation and liquid formulations as well, that include dust, granules, pellets, slurry, etc. So many different kinds of formulation can be and have been developed according to the needs and to the application, to the field application that is needed for this fungus. The fungus is very let's say, easy to grow. It's a fast-growing fungus. The fact that it has been and is being found as a predominant species or predominant genus in different environments means that it's actually very, it can grow very fast and can compete with the other fungi, which limits also the contamination. And several systems, both on solid state fermentation and liquid fermentations, are available, and this leads to the possibility to produce talc formulation, for instance, pellet formulation, or liquid formulation as well, according to the needs. Of course, as for any other biocontrol agent or living organisms, the dry formulation are preferred for their long shelf life. The shelf life of a talcum powder or a granule formulation may last up to 1 year, 1.5 years, although usually it's preferable to use this kind of Trichoderma strains, commercial products, within the 6 months or maximum 1 year from their production. The talc formulation is also very interesting because it allows a progressive drying of the biomass, and this induces the formation of chlamydospores, which are very important for long-lasting and vitality of the fungus in also dry or harsh environments. Another very interesting application for especially in our pastoral system or sylvo-pastoral system, for instance, for the use in olive trees or also urban trees, are the liquid formulation that are based on spore formulation, can be applied directly after pruning, so that they colonize very quickly the surface of the trunk that has been cut, the wounds, and this prevents the infection by other plant pathogenic fungi. So this is very useful formulation as a preventative system to avoid the infection by other basidiomycetes that can then hamper the stability of the plant, of the tree. Another important fact linked to the fact that Trichoderma is very fast growing and able to compete with other microorganisms is that this allows also formulation, the preparation of formulates in low-income countries or poorly equipped with technological means, so that it can be produced even at in very simple ways. Although the picture on the bottom is not related to Trichoderma, is related to Beauveria bassiana, which is another entomopathogenic fungus very well known, and it shares with Trichoderma also the ability to grow very fast in very simple way, can be produced a biomass, a high proportion of biomass, and this allows also the production in low-income countries, for instance. And there have been several experiments on this and several positive experience in India, in South America, in Africa, sub-Saharan Africa, that allowed the production of this fungus even at a very low technological input. The problem here, the major problem, this is based on my own experience, is that there is a need to preserve the inoculum. So the inoculum should be one use only. They should be used only once to produce the biomass, the biomass patch, and then they should be thrown away, because the risk is that the inoculum, the first inoculum, the starter, will get contaminated, and then instead of producing Trichoderma, you produce anything else, which is not good for biocontrol. It's not good for the activity, because you don't have anymore the fungus that you are aiming to produce. We have a long time experience on Trichoderma, because we started our studies on Trichoderma about 20 and more years ago, through a cooperative interaction with a private company, which is the Isagro Biofarming, now Go1 Isagro. And they sponsored us for a research on the isolation of Trichoderma from different environments in Sardinia, undisturbed ecosystem mainly. And they were interested in developing at least one isolate of Trichoderma which could have positive properties, biological control properties, and be commercialized by this company. So we started together with my friend and colleague, Virgilio Balmas, who is connected here, and also another former colleague, associate research assistant, Barbara Scherm. Dr. Safia Fensou was not born yet, probably. Anyway, she was not part of our group. We started a long list of sampling in different sites across Sardinia, including sylvo-pastoral lands, forest lands, grasslands, and also coastal environment. And we were used routinely to collect about 1 kilogram of soil from different places by localizing the sampling site, of course, mixing this soil and then use part of it for trichoderma isolation and part for psychochemical analysis. This is very important. Any isolates should be monosporic isolate. So this is paramount in order to achieve a proper identification of the isolates. So from isolation or rough isolation of different substrates, both PDA, potato dextrose agar, or trichoderma selective medium, we were then collecting the trichoderma looking like colonies and isolating monospore isolation in order to collect pure isolates and to do afterwards a molecular analysis in order to identify the species. And at that time, it was possible to use TrichoKey, which is a very useful website that is now no more operating, but probably it will be operating very soon. So we are struggling in order to make this system operating again. It was very useful indeed. In total, we isolated over 450 isolates of trichoderma from different sites across Sardinia. And this is a general graphics representing the main results of our study in terms of ecogeography of trichoderma. And as you can see here from the pie charts, in each site we found a very different combination of trichoderma species. And there were some sites, like for instance here, EG7, there were a multiple number of species that were isolated. Also here, you can see here. But there are some sites where we found only one single species. For instance, here, I don't know whether you can see my arrow. Anyway, in the northwestern side, we found only one single species, which is Trichoderma gamsi, which, by the way, was not yet described as a species. It was described during the preparation of our manuscript. So this Trichoderma gamsi has been identified mostly based on the high number of strains that we provided to the discoverers of this new species, which was named in the honor of Walter Gams, a very famous taxonomist of trichoderma. Anyway, the fact that a single species within a soil, only one species was found, testifies the great competitiveness of this species. And in fact, Trichoderma gamsis turned out to be a very, very effective biological control agent. So we started a lot of screening in greenhouse by using a model system which was made of faba bean and Rhizoctonia solani. And we wanted to select the very best trichoderma isolates in order to really make a very tough screening among these over 400 isolates that we got. As this is just an example of the kind of results that we could get. As you can see, in some of the lines, there are very few plants emerging, while in other lines, all plants were able to emerge in the presence of Rhizoctonia solani, which was the pathogen that we've been using. And here you have an examples of the selective pressure that was employed, which was very tough. The treatment with Trichoderma solani, with Rhizoctonia solani, sorry, didn't allow the emergence of a single plant. Okay, so this is very important. When you get under this selective pressure, you get this kind of results, means that the trichoderma that we have added to the mix, to the soil mix, is very, very effective in protecting the plants, besides the fact that Rhizoctonia is present. So this picture testifies the very efficient activity of trichoderma, which was able to protect the plants from the attack of Rhizoctonia solani. On the left, you have the tolclophos methyl chemical control, and two of the best isolates in the middle, and a very poor isolate. So this is a very important concept. The fact that trichoderma is not a homogeneous species of genus. So you have many isolates with very, very different activities. Some isolates are very effective, other isolates are not effective at all. So you need a tough work and a very selective work of screening in order to find the best isolates. This graph represents the main results in terms of reduction of the efficacy of the attack of this disease by Rhizoctonia solani. When the disease is 100 or 90, it means that all plants were died. No plants were able to emerge. So as you can see, among several isolates here, there are more than 50 isolates, you get some isolates that were very effective, like this one, for instance, or this group of 3 isolates, or this number 49, and the majority of the isolates were not effective at all. So this again adds the importance of a selection in order to find the best performing isolates. And this applies for biological control, but also for the protection of plants under drought and salinity stress. In our case, we are currently testing several strains, about 20 strains, by using Medicago sativa alfalfa as a model system, by growing alfalfa and try to see whether some isolates are able to provide this increased growth response that was mentioned previously by Dr. Ufensu. And in fact, some isolates seems to be effective in improving the size and probably also the root size of the root biomass of the inoculated plants. So this is a very preliminary test that we are carrying out within the Salam Med project, and we hope to get these encouraging results under practical conditions. Coming back to the agreement that we had with the Isagro biofarming, after about 5 years of work, we came out with a very effective isolates of Trichoderma gamsi, the ICC 080, which was registered in 2013 by EFSA, by the European Food Safety Authority. And through this isolates is one of the 2 components of, along with another strain of Trichoderma asperenum, which was developed by another laboratory in Pisa by our colleague Giovanni Vannacci, now retired. And the 2 isolates, our Gamsi and the Asperellum, are at the basis of a formulation, of a series of formulation of Trichoderma by Isagro, which is now Go1 Isagro. And they are producing and selling all over the world a wide range of Trichoderma-based formulation that are able to improve growth response, to treat the ESCA disease, for instance, on grapevine. ESCA disease is one of the most relevant diseases nowadays on grapevine. And by distributing this trichoderma after pruning, you can get a full protection of the plants. Other formulation also based on these two isolates of Trichoderma gamsi and Asperellum are used for turf, protection of turf grass. And we know that many pathogens of turf are the same that you can find after in pasture land. So although there are no examples that I can show you, but I'm sure that this kind of formulation can be also used to improve the growth and the establishment of pasture land and grassland. The Indian, just to finish this presentation by providing a very quick example of how the research on Trichoderma has been advanced, especially in some countries like India, who have pushed very strong emphasis on the research of Trichoderma. They sponsored it, and the results are really, really visible now, because India is the top leader in all over the world for the production of commercial formulation based on Trichoderma. And there is a panoply, a very large number of companies, private companies, that are producing this fungus or different isolates of this fungus for many, many, many different purposes. So this is an example on the fact that solutions like the living labs, there are fostering business opportunities because the business is there. There are many companies in India that are selling, developing new isolates. They are searching, they are approaching us, also our laboratory, in order to see whether we have some effective strains to be developed. So the market is there. There is a high potential. We know that not only in Europe, but all over the world, there is a tendency to improve and to ameliorate condition within the the principle of conservative agriculture, etc., biological control. So Trichoderma can really find a place, a very important place in this approach. And with this, I think it's the end of this presentation. I thank you very much for listening at us, and we are ready for any questions that you could have or curiosity.
Thank you, Professor Micheli. Very interesting, from the lab to the market. So this answered one of my previous questions to Safa. So I got more questions, but first I would like to see if there are questions from the other audience by Mr. Webbe or Mr. Ahmad Safi. If you have any doubt relating to your own country or the applications in this field, feel free. Otherwise, I have a question I would like to ask. I saw that you sampled the strains of Trichoderma in several sites in Sardinia, and you developed these commercial applications, or willing to develop them. My question was about the issue of cross-border, of using strains coming from one site in another country. Are there issues regarding the cross-border transfer of strains, or are they so ubiquitarian, as we've been told, that there is no issue there? Should the build-up of commercial formulations stem from a collection in one specific country to be used in that country? For example, sampling in Lebanon to be used in Lebanon. So this
Mm.
issue in terms of also of safety, of risk of
Yeah,
contamination.
this is a very good question, Sarah, because it allows me to also to enlarge this this concept. I will give you an example. In the late 90s, I was member of a cost action on biological control formulations and so on, and the impact that this at that time we were still thinking about genetically modified microorganisms to be used in biological control. Then we know nowadays that biological control does not allow the application of genetically modified microorganisms, although I think that in many cases they could be a very environmentally safe solution, especially to reduce the impact that native strains or wild strains, let's say, not genetically modified, may have on any ecosystem. If we introduce a highly competitive microorganism, regardless of the fact that it is genetically modified or not, it has an impact. And therefore, when among the different preliminary experiments that are part of the dossier that any company who would like to register a product based on a microorganism should present to the EFSA, there is the survival curve. So you have to demonstrate that the strain, once introduced in a given environment, the population tends to decrease and to disappear in the end, to get into an equilibrium with the local microflora. The example which was made in the 90s was about Rhizobium leguminosarum. At that time, the first formulation of Rhizobium were introduced in Spain and in France. And there was a very interesting study that demonstrated that 2 years after the introduction of a commercial formulation of Rhizobium, the biodiversity of Rhizobium population was zero, was flattened. So they got 100% of Rhizobia were the commercial Rhizobia. So, and it was not a genetically modified Rhizobium. So it was really a wild strain of Rhizobium leguminosarum, which was very effective, very competitive. And in fact, in several areas where it was introduced, there was no more local Rhizobium present. So this is an issue that is always intrinsic to the application of biological control agents or any microorganisms. If we want to have an effective strain, we have to cope with the fact that the separation between a potential risk and the benefit is very thin. To answer your question, this is a premise. To answer your question, when a product is registered, you get the extension to different countries. So far, as far as I know, there is no limitation concerning Trichoderma because it is considered as a ubiquitous fungus, and it's a non-pathogenic fungus as well. So once you got the permission to sell it to Europe, you can sell it also to other countries, unless there are some local regulation that does not allow this kind of application. But as far as I know, there are no such limits for Trichoderma-based commercial applications. In fact, I saw for a couple of seconds Theodora Fetzi, but she disappeared now. She's a FAO representative in Salam Med, and they are also in FAO. They have these farmer schools, and they were interested in developing such Trichoderma strains also in Egypt and in different countries. And we also offered our collaboration and I renew this collaboration to all our colleagues that are online here, because if they want to apply such formulates, either I think the best way would be to apply a commercial formulates and to evaluate the efficacy of these formulates also in different environments. I think that this is feasible. And last year I got also the availability of the selling company to the Isagro company to provide this some aliquots of this product for free in order to set up experimental trials in different living labs in Northern Africa and all the MENA countries.
Thank you, Professor Micheli. I think there is a question from Salam. Salem Hayar, is that correct?
Yeah, thank you so much for allowing me to ask this question, and thank you for the participant and for the interesting seminar given by Migueli and Safa. So my question is I have double question about, first of all, about the inoculum amount. Up to you, how many the amount of inoculum to the soil, and it's varied from soil to soil upon the type of the soil.
Safa, do you want to? Okay.
Yeah, actually, it depends of the on the soil. For the moment, in our ongoing experiment, we are using sand soil as principal component, and we are using a concentration of 6 colony forming units of Trichoderma.
And the volume? By surface, I mean. How many liquid by surface?
It depends on the volume of soil you are using. For example, for us, we are using small amount of soil. It's about 20 gram, applying 1 ml for 20 gram of sand soil.
It depends also which kind of inoculum. If it's a spore suspension, you can dilute it as much as you can, as you want. I mean, according to your needs. And usually the commercial formulation are very, very concentrated for the reduction of volume, of course. So you can get 10 to the 10th or 10 to the 9th spores per milliliter. And then you have to reduce, of course, by diluting it into water. In many formulations, especially liquid formulations, the company suggests to let the to mix with water and then incubate for about overnight at room temperature in order to allow spores to germinate, to pre-germinate, and this increase the efficacy of the inoculum before spreading it.
But about because our colleagues now was talking about the gram by soil. But what about if I'm talking about 1 hectare, for example? How many amounts of colony forming units you will apply by surface, not by gram?
Again, it depends on the kind of application, because if you want, usually the best way to apply this kind of antagonist is not a field application, direct field application.
It's by drip irrigation. By drip irrigation, yes?
Either by drip irrigation, yes, or at the nursery level. Most of this kind of, for instance, before when you have the seedlings, you inoculate the seedling pods, and then the trichoderma is able to colonize the roots and to follow the life of the plants for all the life, because it's established in the root system. And this is also the best way, because otherwise it would be too costly, too expensive to apply this kind of formulation. You cannot use them as a chemical product. So either by seed coating or by applying it, activating the rooting substrate, or by inoculating the seedlings after emergence.
And up in you, when you apply a fertilization program, it will survive, or I have to apply later after applying the fertilizer to maintain?
Yes. No, usually they are resistant to this kind of compatible with fertilizers. Of course, it depends also on the kind of fertilizer. If you have ammonia, for instance, that could be a bit toxic also for trichoderma. But a standard fertilizer based on nitrogen. Not ammonia, but other kinds of nitrogen, phosphorus.
Because what about calcareous soil, for example, at pH 7.5? It will survive because there is always a fail compatibility between pH and some trichoderma commercial product.
Trichoderma is I mean, it likes low pH, low to neutral pH, but it grows also well. At least some isolates are able to grow well also on alkaline soil, but usually they prefer subacidic soils as a fungus. Yes.
So what is your recommendation for if you have calcareous soil, pH 7.5 or 7, like here in Lebanon, mostly are calcareous soil?
Yeah,
that is the problem of efficiency of this trichoderma.
It can be a problem unless there is a sufficient amount of organic substance in the soil. Yeah.
Okay. Now I have another volley of question about the registration of the product within the Ministry of Agriculture. We call this autorisation de mise sur le marché.
Oui.
Okay. Apenu, is there any harmonized guideline to register this pesticide within European Union that we can follow here in Lebanon, or is there any guideline, any specific specification to register this product? And if we are working on it,
now,
as developing formulation here in Lebanon, what is the guideline here to follow in order to have the registration of this biopesticide, like in Europe, for example?
Do you mean for isolates that you are developing?
Yeah, we are working on isolation. We are testing in laboratory, and we want to move for registration within the Ministry of Agriculture.
Mm-hmm.
That's why I'm asking if is there any guideline to first of all maybe conduct a trial and field, then to move for the registration of the product, like in other country in Europe, for example. Which guideline you are following to register this biopesticide?
Yeah. Okay. First of all, we did not register ourselves a product. This was done by the private company, and this is why we had no forces, because it is very complicated. It's very expensive as well. You need a powerful amount of energies, because you need it's not only a matter of efficacy trial. This is the simplest activity. The problem is that you need to provide chemical all the chemical substances that are produced by the isolate, the stability in different environments, the survival, their potential growth at 37 degrees, for instance, because then it could become a pathogen to humans or to animals. You have to provide data on the toxicity on larvae of insects, fish, frogs,
And
any
yeah, very
kind of
complicated.
yes, it's very, very complicated. I mean, the dossier to produce is really, really a huge one. Unless in Europe, luckily enough, since about 10 years, they have introduced this concept, there is a so-called familiarity. I mean, some species, if there is already a product that is based on, let's say, Trichoderma artianum, that has been registered, all the next formulation based on Trichoderma, other isolates of Trichoderma artianum, have a sort of fast track because there is already a knowledge, a familiarity vis-a-vis of the Trichoderma artianum species. Because it is known that normally this species does not produce toxic substances, etc., etc. But still, you have to provide a very high number of informations concerning the specific characteristic of the isolates that you are developing. And this is why in most of the cases, there must be a cooperation between a scientific laboratory or research laboratory and a private company, because the scientific lab cannot afford such a big
Exactly, yes.
yeah.
But you don't think that there is a way on dit en français, pour alléger les conditions imposées par le enregistrement du pesticide. À quoi sert, for example, to check the toxicity against fish, since I want to apply directly to soil and I'm it's too much complicated test. There is no relationship between the use of the pesticide, biopesticide, and the application and the field.
In the European Union, they did a karakiri.
Here we follow the same. We are complicated here in Lebanon. We are following the same regulation in Europe. That's why you are so difficult.
Yeah, to register a bioproduct in Europe takes about 5 to 7 years, whereas in the United States, for instance, it takes 2 years, 1.5, 2 years.
Mm-hmm.
And in India, I guess it will be even easier. I think it is very important that we simplify at the European level the procedures, because so far is too complicated. It discourage the companies. In fact.
Exactly.
Discourage the company. I agree with you.
Est-ce qu'il y a un programme for the trials? Est-ce qu'il y a is there any guideline to make trials in order to be accepted later by the FAO that your product is efficient or not? For example, there is OECD. Sometimes I see an internet guideline to conduct trials. Is there any specific guideline for biopesticide like Trichoderma or others?
Yeah. I'm quite sure that EFSA has published some guidelines in order to remplir le dossier, to fill up this dossier in order to apply for a registration of a new product. This is EFSA, European Food Safety Authority, is the
Thank you.
the source of this information. I don't know, honestly. I don't have this answer.
Okay, thank you. I take too much time from your presentation.
No, it's interesting. Thank you. I mean, the best way would be to test some commercial strains that are already available in the market and see if it works. And then if there is a positive, and then it is convenient to try to develop new isolates and new effective strains.
Let me share with you some trials that I conducted within my university.
Sure.
I make many experiments with my students regarding the commercial Trichoderma commercials here in Lebanon from mostly countries. Mostly they are not so efficient. Why? Because it's calcareous soil, pH not adaptation. And if really there is a microorganism, bacteria like that, it will not work. Trichoderma will not work. You have to make what you call vide biologique. If you decrease the number of bacteria, other microorganisms, in order to let the Trichoderma launching multiplications and give effect like that. That's why I'm working, and I don't know if you are agree, on some strains isolated from our soil. Maybe Trichoderma selected from our soil more, and if you work on the efficiency on the multiplication more, will be more efficient than the Trichoderma imported from outside. I don't know if you agree on my opinion.
I do fully agree with you. I do fully agree with you because, as I told you before, there is a great variability among isolates, and this variability applies both to efficacy, but also to the ability to cope with specific conditions, soil conditions, environmental conditions that are variable. So if you isolate from the very, it's like the, how do you call it? Personal medicine, right? Individual medicine. That you have to find the right isolates for the specific condition.
What is the best formulation to be used upon you? It's the liquid formulation, the spore formulation, or the solid formulation coated? Which one gives the best results?
It depends on the kind of problem that you want to cope with. If it's to protect plants from soil-borne pathogens,
yes,
it's always best. The principle is that the first who comes, the best it can establish. So when you, for instance, you treat the soil, maybe you do solarization, and after solarization, that's the best moment to include Trichoderma after solarization, so that the soil is like there is a biological vacuum, at least in the
exactly. It's
surface.
the best results that I take in the field after solarization. So to reduce the amount of consortium bacteria and to let then the Trichoderma to be launched and have more efficiency.
Yeah, definitely. And if you want to apply it in nursery soil, for instance, you need to disinfect the soil either with steam or other means, and then inoculate Trichoderma. And that's the best way.
Exactly.
Yeah.
Okay, thank you.
Can I say one thing to?
Yes, Virgilio, please.
Just an opinion for your question. No, the risk to introduce Trichoderma in another country. No, there is a problem, of course, of biodiversity, because if it's a great competitor but we have to thinking that we put this Trichoderma in agriculture soil, so it's already destroyed by the biodiversity and by microorganisms. So it's a little less damage. Okay, what I want to say, just this. I don't know if was clear my English, but
very clear, very clear. I was wondering, in fact, as Forestas, we manage several forests and protected areas, and in some protected areas we have the conservation plan, Piano di Gestione del SIC. It's a management plan that prevents any introduction of any foreign species, being a tree species or a herbaceous species. So again, my question was, and then I'll leave you as we've kind of surpassed the timing of the webinar, but this last curiosity was about the introduction of these strains, because Professor Micheli said that nowadays there is no limitation in terms of regulations from a law point of view, as long as the formulation has been approved for commercialization. So in case we want to use it in a protected area where the plan forbids the introduction of exogenous or non-autochthonous species, does this apply to your knowledge only to plant species, or it should or could apply also to fungal strains? How should we behave in that matter? Should we do a test, a pilot, or use it for biological control purposes?
Quirico, pensaci tu. Sì.
No. When we first did this survey on the Trichoderma species present in Sardinia, we were a bit disappointed by the relatively low number of species that we got in Sardinia. And in fact, the idea was that Sardinia, being an island, would be able to host a very diverse, since Sardinia is a hotspot of biodiversity, a very diverse range of species. Probably we were not, we were disappointed because of this preconceived idea, but in fact, Sardinia likely has been colonized after by foreign species, or we may say exogenous species of Trichoderma later. I mean, after the detachment from Corsica and from France and Spain. Just so geologically, very recently, it has been colonized by exogenous species of Trichoderma. So this is why most probably the diversity of Trichoderma in Sardinia is very poor, relatively poor, compared to other countries like South America, for instance, or many countries in Asia where you have really hundreds of species. We found a reduced number of species in total. I don't think that introducing Trichoderma in a forest environment would determine a change in the microbial composition of this environment, even if it was an exogenous species, because Trichoderma is one of the rich network, and especially in forest soils, there is such a high number of microbials, both fungal, bacterial, and therefore I don't think that Trichoderma could be an issue, even if it was not present before. But of course, maybe a small trial would be advisable if you are not sure, if we want to test the survival capability. But to do this, you would need, for instance, I don't know, metagenomic analysis, quantitative metagenomic analysis before and after the introduction to see whether Trichoderma is there, but the other components are decreasing dramatically or not.
Mm-hmm. So it should be the topic of a specific project, research, an innovation project, yes.
Good. This is interesting.
Why not?
Anyway, well, these species are present in Sardinia, so you don't have to bother. Especially Trichoderma gamsi, it was found all over Sardinia, more or less.
So for Sardinia, we have no problems.
No.
So it's interesting to export these technologies in countries. I'm sorry that there are no representatives from Jordan today. I'm sure there was an issue with getting the Zoom link. So we will verify this. So my apologies for this limited number. So
my message for our friends in Jordan is that, and also as well as to Dr. Hayar and Dr. Safi, that we are willing to cooperate. So if you want to, for instance, if you have strains, isolates that you want to identify, we can try to identify them. If you want to test some formulations, we can make sure that Isagro, Go and Isagro will send you some formulation, some samples to try. We are like, we like collaboration in any case.
I'm really willing to cooperate, and we can sign directly MOU with the School Doctorate of Lebanese University.
Sure, sure.
Okay, and actually I'm working on the yeast.
East of Lebanon. East.
Yeah, yeast.
East, pure noi.
Yeah, I'm selecting now yeast from grapes in order to make mass production and to inoculate again to the grapes.
Ah,
and I still have some, but my always problem is mostly the formulation. It's the secret that I cannot till now. I have a lot of expertise in formulation and chemical pesticide, but not in biological pesticide, microbial pesticide.
Okay. Okay.
So
Well,
let's hope that this contact is the opportunity for starting a cooperation in line with what Living Agro is trying to do to build these. We have a new registration now, so.
That's right. We have a new participant which came in right now.
Good morning, Mr. Abed. Maybe there was some misunderstandings concerning the timing. The time, but also I think there were problems with receiving the Zoom links, because I sent some emails and some people did not receive the Zoom link. So there is an issue with the ICT platform. So I'm deeply sorry about this. We could not discover it before the event itself. So I'm sorry about this. But you've got all your contacts in the chat. Both Professor Micheli just added, and Mr. Hayar, Mr. Canari. So we will also, please note that on the ICT platform, within the catalog of innovations, there is an innovation sheet about Trichoderma that was presented in Jordan during one of the brokerage events. So this has been exported in Jordan, and I think that the contact reference was back then Professor Rogero, but Professor Micheli works within the department steered by Professor Rogero. So also on the ICT platform, besides this chat, you have all the contacts or some information concerning this type of innovation. So should you not be registered, maybe just go and have a look. I will put the link here in the chat. There it is. So thank you so much for the speakers for all these interesting presentation. I wonder if Mr. Abed, who just joined, want to say hello and maybe tell us if he had any problems with the timings or with any issue. But we will be available through these contacts for any further query. Sorry that this has just finished when you joined. So again, thanks to Professor Micheli, to Dr. Safa and Carla Cesaraccio, who had to leave us. Thank you so much to everybody, and please keep the link with the Living Agro project and keep up the cooperation with these countries. Thank you again.
Thank you all. Thank you for joining us.
Bye.
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 Bye. Thank you.
Bye. Thank you all.
Bye.
Federica, if you keep the link open, we can copy paste all the addresses for the report.
Yes, we have the registration. I hope that the chat will be included as well. Okay. Thank you so much all.
Bye.
Thank you.
Thank you.
Have a nice day.
Bye. Bye. Bye.
Bye. Thank you.